Welcome back to Acing AP Biology. In today's video, we will be discussing macromolecules. Macromolecules are large organic polymers. Polymers are large molecules that are composed of smaller subunits called monomers. Just remember that monomers are the building blocks that form huge and complex polymers. Mono means one and poly means many. The process of monomers coming together to form polymers is called a dehydration synthesis reaction, also known as a polymerization reaction. During a dehydration synthesis reaction, monomers form covalent bonds with each other. And for each covalent bond, there is a net removal of one water molecule. One monomer loses a hydroxyl group, and the other monomer loses a hydrogen atom, forming a water molecule. After the removal of the water molecule, a covalent bond forms. The process of polymers breaking apart into monomers is called a hydrolysis reaction. The addition of a water molecule breaks the covalent bond between two monomers. A good way to remember this is that hydro means water and lysis means to break down. So, in this reaction, water breaks down the polymer into monomers. The large polymers that make up life are divided into four macromolecules: carbohydrates, lipids, proteins, and nucleic acids. Before we move on, I want to mention an important theme in bio. Structure always dictates the function. Keep this in mind as we talk about the structure and function of the four macromolecules of life. In this video, we will go over carbohydrates and lipids. The primary functions of carbohydrates in organisms are fuel, structure, and storage. Sugars are the smallest carbohydrates. The monomers of carbohydrates are called monosaccharides. Recall that mono means one and saccharide means sugar, so one sugar. Carbohydrates are composed of carbon, hydrogen, and oxygen, and there is always a 2-to-1 ratio between hydrogen and oxygen, so CH2O. So, when the AP gives you a molecular structure and asks you which of the four macromolecules it is, if there is a 2-to-1 ratio of hydrogen to oxygen, then it is likely a carbohydrate. Carbohydrates consist of hydroxyl and carbonyl groups. Glucose is a monosaccharide and is the major energy source for organisms. Breaking carbon and hydrogen bonds release a lot of energy. Each carbon atom in a carbohydrate molecular skeleton is bonded to a hydroxyl group. However, one carbon is double bonded to an oxygen, forming a carbonyl group. Recall that in the last video, we talked about aldehydes versus ketones. In aldehydes, the carbonyl group is on the end. In a ketone, the carbonyl group is not on the end. Some examples of monosaccharides are glucose, fructose, ribose, and deoxyribose. Carbohydrates can exist in a linear or ring structure. Take a look at glucose. Notice the hydroxyl groups and the carbonyl group. Each corner of the ring represents one carbon. Sometimes the carbons will be omitted, so just assume that each corner is a carbon. Two monosaccharides come together via dehydration synthesis to form disaccharides. The covalent bond formed between two monosaccharides is called glycosidic linkage. Some examples of disaccharides are maltose, which is made up of glucose, and sucrose, made up of glucose and fructose. As you can see, when maltose forms, two glucose molecules undergo a dehydration synthesis, where one glucose molecule loses a hydrogen from the hydroxyl group, and the other glucose molecule loses a hydroxyl group to form water. The covalent bond that forms is called glycosidic linkage. Polysaccharides are carbohydrates composed of hundreds to thousands of monosaccharides. They can be divided into two major functions: energy storage and structural support. There are two prominent storage polysaccharides: starch and glycogen. Starch stores carbohydrates in plants. They are stored in plant organelles as granules, known as plastids. Glycogen stores carbohydrates in animals. Glycogen is more highly branched than starch. Glycogen is found in the liver and skeletal muscle of the human body. There are two major structural polysaccharides. Cellulose is the prominent structural element in plant cell walls. Cellulose is held together in bundles of microfibrils. Cellulose molecules form hydrogen bonds with each other. Next, we have chitin. Chitin composes exoskeletons, which are tough external skeletons found in arthropods. Chitin is also found in the cell walls of fungi. Chitin contains an amine group. Lipids are hydrophobic molecules that are insoluble in water. Lipids do not form polymers. There are three prominent groups of lipids: fats, phospholipids, and steroids. Fats store large amounts of energy in organisms. 1 gram of fat stores 3 times as much energy as 1 gram of polysaccharide. This is due to more high-energy hydrocarbon bonds in fats than carbohydrates. Fats also function in cushioning and protecting organs in mammals. Fat also provides insulation. Fats consist of the triglyceride structure, which is made up of 1 glycerol molecule and 3 fatty acid tails. The fatty acid tails usually contain 15 to 16 carbons. The fatty acid tails can vary in length and in the number and location of double bonds. The bond formed between the hydroxyl group and the carboxyl group is called ester linkage. It takes 3 dehydration reactions and thus the removal of 3 water molecules to form a triglyceride molecule. Let's talk about the difference between saturated and unsaturated fat. Saturated fats have no double bonds in the fatty acid tails. The carbon atoms in the fatty acid tails are saturated with hydrogens, meaning that each carbon bonds with the maximum amount of hydrogens. Examples of saturated fat include animal fats like lard and butter. Unsaturated fats have 1 or more double bonds between the carbons in fatty acid tails. These double bonds are called kinks. Examples of unsaturated fats include plant fats like corn oil and olive oil. Therefore, the fat in this drawing would be saturated because all the carbons hold the maximum amount of hydrogens it can because these are all single bonds. Phospholipids consist of one glycerol molecule, two fatty acid tails, and a phosphate group. Due to the phosphate group, phospholipids have both nonpolar and polar regions, making it ambivalent towards water. The head of the phospholipid, where the phosphate group is hydrophilic. The tails, or the fatty acid tails, are hydrophobic. Phospholipids are a significant component of the plasma membrane, also known as the phospholipid bilayer. Lastly, we have steroids. Steroids are composed of 4 fused carbon rings with functional groups attached, for instance, like a methyl group. Notice that these are 3 hexagons and 1 pentagon. Examples of steroids include cholesterol, sex hormones, and bile. Cholesterol has a very important role in the plasma membrane, which we will discuss later. That's it for this lesson. In the next lesson, we will finish up macromolecules, discussing proteins and nucleic acids. See you then. For more bio content, hit subscribe.